Our next speaker is Dan Yamins. And Dan is at Psychology and Computer Science at Stanford University. And I just found out that he's also at the Woodside Neuroscience Institute. And Dan is talking today about a fruitful reciprocity, the Neuroscience-AI connection. So, Dan, it's all yours.
Thank you so much. As Peter mentioned, I'm going to be talking about the reciprocity between neuroscience and AI. And in our group, we work on sort of three basic areas. One is cognitive science, that is essentially, in our case, something like benchmarking humans and machines at scale. Okay? We also look at neuroscience topics, that's essentially making predictive models of brain data. And the glue here is really artificial intelligence. So that's basically building neural networks to solve cognitive tasks and then use those to generate hypotheses both for the cognitive data and for the neuroscience data internally to the models, right? So there's really these steps, essentially thinking of cognitive science as target setting for AI, and then AI as hypothesis generation for thinking about the brain. Okay, I'm going to mostly be talking about AI helping neuroscience today, so the direction of using AI models to make models in neuroscience, although it can go in the other direction as well. Okay, so one of the problems that we work on that's sort of most visually obvious and also at the base of a lot of our work is visual object recognition. So it's basically knowing what is where by looking, okay? And even if you haven't seen this particular scene in Cambridge, Mass before, you know that what you're looking at are these different types of objects, and it's quite robust and effortless for you in domain general, okay? Some of you may know, but I'll give you a little bit of background, that this type of ability is supported by what's called the ventral cortical pathway of brains, primate and non-human, humans and non-human primate brains. And in the ventral pathway, there are a series of brain areas, starting from kind of the retina, and then going up through early visual cortex, that's like V1 and V2, those are ventral 1 and ventral 2, through intermediate visual cortex, ventral 4, and then up through higher visual cortex, that's also known as inferior temporal cortex, and there's different parts of that. The key thought is understanding, though, is that if you ask humans or non-human primates to perform recognition tasks, like knowing what object is in an image, data for being able to solve that is present in the higher visual cortex. So essentially, if you look at neural responses in V4, you get some performance on a recognition task. But if you then look at IT downstream in higher visual cortex, the performance is significantly higher, okay? At the level, near the level of human performance itself behaviorally. So it's like saying these neurons are responsible for that type of behavior. Okay, so that meant, and this has been a long ongoing project in various ways, is that our goal is to build quantitative models in that system. Okay, and starting more than 10 years ago now, we've been using neural network models to do that. An earlier class of models that is still quite powerful for this purpose is convolutional neural networks, okay? So these are basically networks that were designed originally to condense the rough neuroanatomy of the ventral pathway, in that they are hierarchical, meaning they come in a series of layers or sections like I just showed you, and they are retinotopic, essentially, that is to say spatially tiled or convolutional. Okay, individual parts of those layers, the internal structure, is made up of neurally plausible basic operations like linear filtering combined with some nonlinear structures like thresholding, pooling, normalization, each of which could in principle be implemented by one or few synapses. Okay, and you can think of these operations as having kind of neuroscience versions and data science or machine learning meaning. I won't get into the details of all of those today, but that's like kind of summarizing 50 years of research in the field. But the idea is that those operations are applied convolutionally. They're essentially the same at all locations, dragged around the image. And by virtue of doing that, if the image is an input, if you have image-like input, then you end up with image-like output with like one kind of filter, one slice for each filter. Okay? So this is how a convolutional network works in general, and then it's stacked hierarchically to produce deeper and more complex operations. Okay, so that's a hierarchical convolutional neural network. Now, In principle, that makes a lot of sense for thinking about the visual system, since it's sort of based on similar principles, at least at a high level. But of course, there's a huge number of parameters that are consistent with this concept, meaning like both architecturally, like how many layers, how many filters, et cetera, together with more like fine-grained continuous parameters, like the filter parameters, right? And so a big question is how you would discover the right parameters to understand real cortex, okay? There's many ways to do this. But one way that we have found over time to be most effective is basically what we call goal-driven modeling. That is to say, optimizing the network for performance on real-world tasks, like categorization, and then looking at the internals to see if they match data. So this is not direct optimization toward data. It's optimizing for a task that you think the brain system performs, and then you can see how well that constrains the internals. Right? Now, actually doing the optimization is a bunch of work, right? And it's been a bunch of work over years. As we get better and better models, we have to work hard to actually solve the optimization tasks. But assuming you can do that, what you end up with is higher performing models, such as that red bar here, that performs reasonably well on the task. And then you can ask, how well does it describe neurons? And a key early result was that there's a strong correlation between these things. In other words, across thousands of models, that's what each of the dots are, performance on a task, the x-axis, highly correlates with ability of the model with that performance level to predict neural responses internally. Okay, so that's like saying that you can get pretty good models by optimizing for a task, pretty good models of the brain system that solve that task, right? And since that early work, deeper models have been able to go beyond that, and in particular, pick apart very fine-grained differences between areas, such as sort of models that have early layers that look like V1, early visual cortex, internal intermediate layers that look like V4 cortex, and then higher layers that separately parcel out different parts of higher visual cortex, like posterior and central and interior IT, inferior temporal cortex, right? And the key thing here is it's the same it's the same model that accounts for all the areas, right? You don't need a different model for different areas. It's the same model with sort of anatomical consistency, meaning the proper area of the model describes the corresponding area of the brain. Okay, and there's sort of at a very high level, what I'm describing for you here is a kind of complementarity, I guess you could say, where there's a classical approach in neurophysiology of going kind of bottom up, like looking at the features or look at what's computed in the neurons in early layers and going up the system to try to construct an idea of what's happening in the higher areas. What I've been telling you here is that a top-down kind of end-to-end goal-based approach using neural networks does a good job, meaning you impose a task on the system, and that constrains what the system is like internally in a powerful and meaningful way. Okay, and of course, since early work, there's been a lot of further types of models that people have developed, like with anything where you can assign a score, you can get models better, and the Brain Score project has been quite helpful at organizing all of that work and motivating people to produce better and better models of the whole system, okay? But it goes beyond just vision. So here's another related slide that comes from thinking about the auditory system. Okay, so here it's different classes of models with somewhat different architectures solving a different task, but nonetheless, if you solve the task better and better, that's the x-axis here, you get substantially better at having internals of the model look like the data, okay? And the correlations seem quite high here, but we've never been able to break them. Okay, so we think that there is something strong about that actual meaning in that system. And in particular, although these are different classes of models solving different tasks, it's a similar principle. A high variation task performance drives models that look like the internals in the brain. Okay, and this actual approach has, as I say, been applied to primate and human vision, that's been applied to human audition, but actually it's also been more recently applied to things like animal cognition, so animal vision, as well as memory and mouse medial entorhinal and hippocampal system, as well as in the work of many others in the motor system, in olfaction, and actually recently in human language. Essentially, you optimize word models and you show that the internals of those language models do a pretty good job of describing neural responses in the human language system. Okay, and you can ask what kind of understanding this approach is, right? A sort of classical form of computational neuroscience works with sort of a little bit cleaner ideas. So, some kind of closed form formula with provably optimal theoretical properties, and the description use, for example, of trying to describe neurons in early cortical areas like V1 with Gabor wavelet filter banks, okay? So what I'm telling you here is not that, okay? It's not that clean, okay? But it's also not the same as just a purely empirical approach where you would say, take a neural network and just fit it to the data in an online way, right, end-to-end way, right? It's not that either, because instead what's happened is that we've optimized networks to solve tasks and then found that those are highly constraining. Okay, so it really plays on the notion that nothing in biology makes sense except in light of evolution, or the neuro version of that, which is nothing in neuroscience makes sense except in light of behavior. And so we can translate that in our words into thinking that nothing in neuroscience really makes sense except in light of optimization. Okay, or maybe we should say computational neuroscience, but that's the underlying theme. Or in other words, behavior is highly constraining of the brain for animals at their scale and for humans and non-human primates at their scale. Okay, bigger mechanisms, but highly constrained by the harder tasks that they do, right? So underlying optimization models are these kind of 4 basic ingredients. One is the architecture class, so that's like the circuit neuroanatomy, okay? 2 is the objective function, or you can think of that in sort of biology terms as the ecological niche. 3 is the environment, or the data set that it's trained on, and 4 is the learning rules, so the natural selection and synaptic plasticity, right, in biology terms, right? And what I'm basically telling you is that, say, as of something like 2016, okay, the best proxies for the ventral pathway, for using this idea to construct a model of the ventral pathway, the visual system, was to take the architecture class to be convolutional neural networks of reasonable depth, to take the task to be multi-way object categorization, to take the data set to be like ImageNet images, okay, and to take the learning rule to be a kind of evolutionary architecture search plus filter learning through gradient descent, okay? And that's a nice story, but actually it has some ingredients to it that are sort of, while powerful kind of from a predictive sense, obviously wrong, okay? So one is that the architecture class has no recurrence and feedback and no topographical structure, both of which are key features of the actual brain, okay? The task or objective, right, required lots of labeled data, and we know organisms aren't getting that. Moreover, data sets like ImageNet are these stereotyped clean still images rather than real noisy video that actual organisms get. And then, of course, there's all sorts of problems with using backpropagation, and neuroscientists will tell you how non-neural it is. That's a long story. But each of these things is okay, it's a problem, but it's also an opportunity for figuring out what to do next, okay? And so a lot of the work that we've been thinking about in the brain area has been dominated by working toward fixing those problems. So I'm gonna tell you a little bit about each of these in the remaining time that I have, okay? The progress that's been made. So let's start with the first one, which is recurrence as an architectural issue, right? If you look at an actual brain, what you see is that there's lots of local recurrence, and there's also lots of long-range connections, feedback connections in the brain, to and fro brain areas, right? Thinking about that in context, we've made models that combine convolutional structure with recurrence. Okay, work of mine due to my former graduate student, Aran Nabi, who's now doing his postdoc at MIT. And basically what that means is a convolutional neural network that also has local recurrent operations, that's like the green arrow, and long-range feedbacks, those are like the red arrows. Okay, it's a recurrent neural network, but it's one that's essentially got a backbone of a forward sweep, like the kinds of the models that have been previously shown to describe ventral pathway reasonably well. Okay, so what's the computational idea here? The idea is that you can do better without having to be very deeper. So if you take a convolutional feed-forward model of a given depth, it does so well on a task like ImageNet performance. If you double the network depth, of course it's much bigger in terms of number of units, it can get better at that task, okay? But we don't think that's how the brain does it. It's you don't get a lot better at a task by doubling the size of your head case. There's gotta be some better thing, okay? So that's the notion of where recurrence comes in. Now, some of you may be familiar with standard recurrent cells, such as long short-term memory cells, LSTMs or others. Turns out that those are not particularly effective in the context of a convolutional structure. However, with the right structure, and the details of which I could talk about if people are interested, but let me just say are inspired by structures that are observed in the brain, you can actually, with a network that's equally deep to the black bar, produce performance that's about as good as the network that's twice as deep, without having to double the number of units, just a very small number of units, and mostly just back connections and recurrences. Okay, so with that optimized structure where you have some local recurrent network of a given form and some long-range feedbacks, you can improve performance. But can you also improve your ability to predict neural dynamics? That's the question. Okay, so one way to evaluate this is to compare what people call easy versus challenge images. So easy images are ones where neural networks that are feed-forward do well, and images are ones where they don't, but where animals or humans still get it right. Okay? And an interesting observation is that in the animal data, this is in non-human primates, if you look to decode the image contents, the challenge images take that animal longer, so like 20 or 30 milliseconds longer to solve. Okay? So meaning, presumably, some additional processing is occurring, perhaps recurrent processing. Okay? So how would you show that? Okay? How would you infer that that's actually what's happening? One way to do that is to notice, okay, each image gets solved at a different time by the animal, and you can look at the object solution time value for each image by image in the animal, and you can also use a convolutional recurrent neural network to make predictions about object solution time, because that network will have times when it solves each image as well. And you can ask, are the animals' neural solution times similar to the solution time pattern of the convolutional recurrent neural network? And it turns out that this is a key result here, is that dynamics fit, that's on the y-axis, is optimized by those convolutional recurrent neural networks, okay? If you look on the x-axis, that's percentage of challenge images solved. Deep neural network, feed-forward networks with hundreds of layers can also solve those images, but they don't fit the pattern of when they're solved nearly as well as the convolutional recurrent network. Okay, so this suggests that the recurrent network, this convolutional recurrent network, are matching the key sort of neural dynamic properties of the brain in a useful way. Okay, and if you think about this from a point of view of why is this the case, and look at model performance versus model physical size, deep feed-forward networks are very big and solve lots of tasks. Shallow feed-forward models are not so good, but shallow-ish or brain reasonable depth convolutional recurrent networks achieve a good trade-off between performance and size. So they're kind of right at this Pareto optimal between being not too big but solving a task. So that really indicates that recurrence is there to make the brain case a reasonable size. It's really basically a biophysical principle. Okay? So, you know, thinking about that doesn't mean we've come anywhere close to like actually completely nailing the question of what recurrence is doing and how it should be involved in these networks. That would be good. We are not there. We're more like in the okay-ish state of having models that are harder to completely reject out of hand. Okay? Better than we were before, being obviously deeply wrong, okay? So that's where I think we are from the architectural point of view in terms of recurrence and feedback. With that in mind, let me move to talking about what I think is one of the biggest issues that sort of plagued me when I started my lab a few years ago and was something which I wanted to make progress on, and lo and behold, due to both our work and also kind of the amazing work in the community in AI, huge progress has been made there, okay? And that is in the issue of supervision. So there's just no way that organisms like this receive millions of high-level semantic labels during training. Like, okay, humans probably don't, and macaques certainly don't. Okay? Supervision might be an effective proxy and might be what's optimized over the evolutionary timescale, but that's clearly not what's happening in terms of the actual mechanics of learning. Okay? Real visual experience is more like this. Like basically babies or toddlers see stuff and they hear some audio and stuff perhaps, and then even if they don't hear audio for one reason or another, they still learn how to see pretty well. And so that's formalized by this very beautiful data set that was collected by, among others, my colleague Mike Frank at Stanford. This is the SAChem data set. This is a data set of a head-mounted, basically they put head-mounted cameras on several toddlers, infants, and they tracked them from 6 months of age to 32 months of age with, as I say, this head-mounted camera. It had mono, audio, and visual channels in it, and they recorded a couple hours a week, right? So, like, the big challenge is, like, how would you use a data set like this to learn, where you don't have labels, it's not well formulated, it's like video data, et cetera, right? So obviously at some level you need some kind of unsupervised learning to do this, okay? But the question is what exactly, okay? Unsupervised learning with neural networks or others has a long history, but basically for a long time it kind of just didn't work that well. Okay, that was basically the state of affairs up until like 2018, 2019, right? So thinking about an autoencoder again, we saw an autoencoder in the last talk. Here's an autoencoder formulation of an unsupervised learning problem, which is essentially image comes in, X. The network is supposed to reconstruct the image, X hat, and is trying to minimize the difference between those two. That's the first part of loss function. But of course, to make that a non-trivial problem, you have to have some kind of bottleneck or penalty in the middle. Okay, in this case, a very classic form of this was the sparse autoencoder that was shown in the mid-90s by work of Bruno Olshausen and others to do a really good job of describing V1 neurons. So basically, if you have a sparse activation penalty on the hidden layer of a shallow neural network, and you optimize it to do reconstruction in natural images, the filters that come out look a bit like Gabor wavelet filter banks, which in turn look a bit like V1 neurons. Okay, so it's like saying neurons have to represent their environment efficiently, right? Ideas like that were sort of limited though, and if you try to apply them to much deeper networks in a naive way, it doesn't actually produce better representations, right? So this field was kind of stuck there for a while. Since then, another idea had come up, which is sort of missing data problems. Like you give me something, you give me the full image, or the data, whatever it is, take away some portion of it, and then use what's remaining to force the network to predict what was taken away. And you hope that the representation that is learned to do that learns something meaningful. Okay, so one of the neat versions of this is colorful image colorization. This is basically where the autoencoder is, instead of trying to reconstruct the original image, you take the original color image, grayscale it, push that grayscale image through the network, and then force the network to be learned parameters that will colorize the image, right? So now that's no longer in danger of triviality because you're not just trying to reconstruct the original image, okay? And the concept is that perhaps you need to learn something about objects to know how color should be distributed in the image. And it turns out that that did a better job, okay? Stuff like that, missing data problems were better than standard autoencoders. But anyway, none of those methods were really all that powerful. And so, starting in like 2018, 2019, a new form of methods came out that really started to change things, and that's called deep contrastive embedding. Okay, so I'll give you an example of an early form of contrastive embedding developed by Stella Wu and some of her colleagues at Berkeley. And this was the instance recognition problem. So basically, here you have a network actually without an encoder. Without a decoder. There's no autoencoder here. It's just a deep network, like of the standard kind, that produces an embedding. And that embedding, and now instead of being optimized to solve categorization, is optimized to put different versions of the same image, push them close together in embedding space, while pushing apart images that are not that image. Okay, it's really simple. I have the same image. I might make general versions of it by cropping it in different ways. It's as if you move your head, okay? And then you push together the embeddings of those while just pushing apart anything else you see. And it turns out that that led to quite an improvement in terms of the internal representation's learned ability to solve other tasks. And it sort of started to kick off a whole new field, among which my former PhD student, Seung-Shu Zhuang, contributed very significantly to. And in particular, he has a whole series of papers developing both contrastive learning methods and also applying those to thinking about the brain. Right? And in his method, called local aggregation, what he did was not only try to push together the same image, but also figure out in a kind of unsupervised way what other images are likely to be similar, while things that are probably different, push those apart by distinguishing close versus background neighbors, okay? I won't get into the details of the math about how that contrastive optimization is done, but nonetheless, the point is that if you start out before training in a kind of randomized way, what you end up with is the development of kind of multi-scale clusters at various, without having to have any initial labels for them. Okay, and this was a really nice point because actually this network was the first unsupervised network to do better than supervised deep nets that had originally kicked off the deep learning revolution in 2012. So that's an arbitrary point of reference, but it just was to say that these unsupervised networks were able to make substantial progress on the problem and push they're sort of coming into their own as actual solutions to the problem, right? And not only do they do solutions to categorization tasks reasonably well, they also solve lots of other tasks, okay? Which is what you'd expect, since they're not optimized for any one particular task. They're sort of task agnostic, and then you hope that the representations generalize. And indeed, they generalize reasonably well to a variety of types of tasks, like object position estimation, object size estimation, and so forth, okay? For us, though, the key question was, how well do they predict neural data, right? If you take the neural networks in this kind of unsupervised way, compare them back to real neurons, what happens, right? Basically using a similar procedure to the one I hinted at before that had been used to think about the supervised networks, right? And this sort of tour de force paper that Cheng-Xu had, he showed a lot of comparisons of this form and really established unsupervised models as the best existing model of the ventral pathway. So in particular, what he found was that contrastive embedding models finally were at a position where they could be about as good, if not slightly exceed, supervised models at predicting neural responses in the brain systems. Okay, and so this was really kind of the first time that there was a quantitatively accurate unsupervised model of a higher brain area. And not only was this something that was true for one particular brain area, but again, there's this kind of anatomical mapping consistency where early layers of the model were best at describing V1, or early visual cortex. Intermediate layers were best at describing intermediate visual areas like V4, and higher layers were best at describing higher brain areas such as IT. Of course, that's all on static images. But one of the original things that I said we needed to be able to do was work on video. And Cheng-Xu was able to develop methods that took the notions in images and bring them into the video context, where now there's static processing stream, that's a standard kind of 2D network, as well as dynamic processing stream. That's a kind of spatiotemporal filtering. Okay, and you can train that network to solve an unsupervised video embedding task, similar in nature to the ones used for images. And it turns out that that works from a computer science point of view, so that's like a computer vision advance. But the real thing from the point of view of our investigation today is to think about how you would use that to apply back to the type of data that I mentioned earlier, the head-mounted cam data, right? And where it looks basically like this, right? And so this is a really hard problem because real kids' data doesn't have the kind of ability to be buffered the way that big data sets do, computer vision databases do. They have many fewer distinct examples, right? You don't see 1,000 different cups or 1,000,000 different cups. You see a few, but you see them from a wide variety of viewpoints, okay? But in any case, regardless of the difficulty of the problem, the key point is that with an unsupervised video processor, you can stick the video of that kind into the actual neural network, okay? And it turns out that if you do that using the sort of contrastive video methods developed by Cheng-Xu, then you end up with networks that have learned to predict neurons as well, if not slightly better, than things trained on ImageNet. Okay, so it's like you can actually use real-world data from the organism to train the network. Okay, and that is a huge difference from, say, earlier visual, like unsupervised video processing methods, such as, like, you may have heard of predictive coding. Simple versions of, versions of predictive coding implemented do quite a bit worse on this, suggesting that, you really need to do something more like contrastive learning to be able to get networks that perform well and learn brain-like representations from real-world data stimuli. And, in the long run, what we'd love to do is make real models of visual development, right? Obviously, the data for that is hard to come by, but that is something we work on with our collaborators. So, the takeaway so far, right, from what I've said is roughly that the contrastive unsupervised approaches sort of finally made up that supervision gap, and they work pretty well in real data streams, okay? So looking back at our report card here, we've looked at sort of gone from like bad to like okay on a few different things. So having recurrence and feedback, not needing labeled data, being able to handle real world noisy video data streams, okay? I'm not going to say too much about the backprop question. I'll come back to that in a minute. But obviously, it's still imperfect. So if you look at this, the dotted line is where the noise ceiling of the data is. So what this is saying is that the self networks are like not solving it completely. right? That would be something that we really still have to solve. But what's particularly interesting to me, and I was sort of chagrined about this when I sort of realized what this meant, was that I kind of expected, okay, if you can really do better with unsupervised approaches, you ought to be able to do better than supervised ones at describing neural responses significantly. But actually what you see here, at least on this metric, okay, is about as good. But it's not better. It's not like something that's more natural in its learning process is like that much better at describing the neurons. Right? So that was something that sort of made me think, okay? And so now I'm going to tell you another story that ends up being highly related to this one. It didn't obviously start that way, but it will end up there, okay? So I'm going to take most of the remaining time to focus on that, okay? So you might think, what do we know? This is something we thought about some time ago. What do we actually know about the visual brain, okay? On the one hand, we know about neural response properties. That's like neurons and how they respond to stimuli, okay? Everything I've talked about so far is kind of in that category. We also know about neural arrangements, okay? Like literally neurons are on, so the surface of the brain, the cortex is on the surface of the brain, right? And like the neurons are arranged on that surface in like a very stereotypical physical pattern, okay? You can think of that as sort of topography or functional organization. Okay, and in the visual system, there's lots of very characteristic functional organization or functional topography. Okay, one, for example, is that in early visual cortex, what you have are maps in various different dimensions. So if you look at humans, non-human primates, a few other species, what you see is these characteristic pinwheel representations when you tag neurons for the orientations that they like. V1 neurons in early visual cortex like different bars of basically different orientations, different edges of different angles. And if you tag them for their preference, what you see is this kind of interesting pattern where there are these large iso orientation domains spatially with like sort of interspersed with singularities in the so-called pinwheels. Okay, that's what those colors are. And if you look at things other than frequency, than spatial orientation preference, you can also think about like spatial frequency or color responsiveness. You get maps of various kinds. Okay, so these are spatial maps that are present, functional organization that are present in early visual cortex. In higher visual cortex, there's a very different, or at least apparently very different form of spatial organization. So in this case, it's like patches of neurons that seem to respond to categories of various kinds. So like faces or places or bodies or, organized according to things like an axis of animacy or other things that people have described, real world object size. Okay, so there's organization of higher visual cortex according to various topographic structures. Okay, apparently quite different from those that are present in earlier visual cortex, right? And these observations are like among the most robust things that are known in neuroscience, right? So our hypothesis sounds like it's totally unrelated to unsupervised learning. That's going to come back. Is that regardless of how you solve this problem, there is an underlying combination of solving a task and optimizing some kind of biophysical constraint, and that should explain all of the topographical data. Okay, so just to make that more concrete from a neural network's point of view, imagine you have a neural network solving a task with its loss function, part of its loss function, and the other thing that it's doing with its loss function is minimizing some kind of biophysical or spatial cost. I'm sorry that's cut off at the bottom, but it says total loss on the system is the categorization loss plus alpha times the spatial loss. Okay, so alpha is like a weight that says how important the one component of the loss function is relative to the other. The biophysical cost versus the task cost. Okay, and so the question was, could you generate patterns of all different kinds with the same model in this way, in a unified fashion, right? To do that, though, you have to actually add something to convolutional neural networks, or neural networks in general, which is that although there's sort of spatial retinotopy, the channel dimension, the dimension of the different filters in the network, or the different neurons at a given spatial location, are not laid out in any spatial way, right? Conv nets or other deep nets don't have any claim on that, okay? So to make it connect to the problem, you have to lay them out. So basically, putting the neurons in an actual physical surface, in this case it's a 2D square, but that's sort of meant to be like a tissue model, a spatial model on the surface of the cortex. Okay, and you can do this for every area, and this is how people see it. You see these sort of retinotopic fields laid out spatially separately. Okay, so this is to model that idea. okay? And then on those fields, you can apply spatial loss with performance optimized at the end, right? And so the question is, if you do this, what happens? Networks of this kind, I'm going to call them topographical deep artificial neural networks. And you can ask, do they predict the topography of the real system? And the basic straightforward answer is they do very well. So in V1, when you want to observe orientation maps, what you do is you probe the system with drifting gratings, bars of different orientations basically, and then you measure orientation tuning and make a map of the preference. So it's quite hard to know whether this comes from data or an actual model. This turns out to be a model. That's at the 1 centimeter scale is that box. You have to pick and fix a size scale for the units to be able to make a direct comparison. If you blow up to the 1 millimeter scale, what you see is that the networks are not totally ordered, but they have exactly the structure that you might expect from the not totally ordered data in the real system. And you can see that both for orientation tuning, as well as spatial frequency maps, color preference maps. That's a cytochrome oxidase stain that basically says whether the cell is responsive to a certain kind of color regime. This is qualitative. You can also quantify this. We could spend a long time discussing exactly what the proper metrics for matching neural network maps to data maps are. Suffice it to say that we believe that we've done that reasonably. And when you do that, you find that the sort of orientation tuning, spatial frequency, and color maps of these topographical deep networks do a very good job of describing the actual data in macaque. Okay, so they're pretty smooth, they've got pinwheels, but they're not as smooth as they could be. And actually some earlier models made things that were very smooth and actually much too smooth to actually describe the true data, as well as these better, more recent deep neural network topographical models. So something about the data is like a little bit dirtier than you might think from a very simple, clean perspective. But whatever that weirdness is well captured by the actual topographical neural network. Okay. So that's V1-like functional organization. That's early in the system. You can also ask about higher visual cortex. So in macaques, that's called IT. I'm slipping in here. Then in humans, that's called ventral temporal cortex. That has a slightly different name and position, but those are homologs between the human and the macaque brain. And so you can ask, how well does that do, right? And that can be quantified in a lot of ways. Actually, this is a place where the field is still struggling to figure out how to quantify exactly what the structures are. There's not really lots of good metrics for the presence and spatial structure of patches and things in higher visual cortex. But if we've assessed a bunch of metrics, including like patch existence and selectivity, and we find that topographical deep neural networks do a good job of describing those structures as well. So you can also look not just at like the existence of selective units, but also at the spatial relationships and size of the patches. And what you find is then there that topographical neural networks do a pretty good job of describing those patches as well. And in particular, you can drill down into things like the number of patches and the size of those patches in terms of their area on the cortical surface, as well as the interrelationship distance between patches of various kinds. Okay, so right now, these models are not perfect, but they're very much our best existing models of the topographical structure in higher visual cortex, right? So with the notion in mind that those models are doing a reasonably good job of describing the phenomenology, okay, or emerging the phenomenology, right, because that phenomenology wasn't exactly built in. With that in mind, what can we say about the structure of the network and what had to be true for that to be the case, right? So I sort of implied earlier that the network that did this was one that was optimizing for the spatial loss as well as for categorization loss, right? But actually, the network that I was showing you was the self-supervised network that came from the earlier picture. We started with the categorization-based network because that's what we had at the time that we started this project. Okay, but along the way, this other one came out. We've developed that other thing for sort of a different project. And it turned out that that made a big difference. So both in early and late visual cortex, using the self-supervised network, that's the one that ends up being part of the TDAN, that topographical deep artificial network, made much better matches to topographical structure. Well, somewhat better structure in early visual cortex and much better structure in higher visual cortex. Okay, which is interesting because you'd think a categorization network would be better at having category selective patches. But the problem is that ImageNet kind of gives you the wrong distribution, whereas the self-supervised network actually gives you the right selectivities to match the real visual structure. Okay, so another way to say this is that, like, unlike if you're just looking at the neural distributions themselves, where self-supervised and category-based networks are kind of similar, for these topographical metrics, the self-supervised networks are actually a much better account of the real system, suggesting that actually that better realism in the underlying learning mechanism is really impacting the ability of the network to describe the biology, okay? Of course, you might wonder why there is a spatial component at all. Why do this, okay? Well, okay, another way to, like, I'll tell you the like quantitative result, but which is basically that if you measure wiring length of the system, this is kind of a biophysical cost, and you look at it across the spectrum of the value of that alpha parameter, how much does the spatial cost matter, what you see is that at the place where actually all of the biology emerges, that's around alpha of somewhere between 0.5 and 1, okay, that actually is the one place that minimizes the wiring length. Okay, so that's another way of saying that in kind of technical terms, is that the loss circuits are optimized to support general task transfer learning, that's the self-supervised learning component, and minimize feed-forward wiring length while doing so. Okay, so just to sort of think about it sort of physically, imagine you were spray painting a computer onto the surface of the brain. How would you organize it chip-wise so that it was able to do a task, but have the wires be organized well, right? So they aren't too long. That's what this is saying is occurring in the system. Okay, obviously that's not a metaphor. That's not an exact procedure for building spray-on computers, but it suggests that if you were to move toward a more physical implementation and biomimetic structure for that chip, you really need to take into account wiring length in this kind of like biophysical way. Okay, and actually similar ideas can explain much higher levels of data. So I was just telling you about the ventral pathway, but visual system is broader than that. There's actually multiple streams in the visual system. Ventral, that's sort of the bottom part of the brain. Parietal, that's sort of across the top, and lateral. Okay, and people have wondered for a long time, why are these systems separate? Of course, in some ways, the predominant theory is that they're there to, these systems, these sort of pathways are there to accomplish different tasks, like object recognition versus action recognition, or what versus where, or lots of other things, okay? But actually, there's an alternative that's suggested by what I just said, is actually the whole system is just spatially organized for some single task, right? That's a quite different theory. It says, okay, there might be specialized subareas, but they emerge just by having one system laid out in space, right? And so that's like saying, hypothesis one is there's behavioral demands. Hypothesis two is spatial constraints. That's basically just the single TDAN model that I mentioned earlier. And I won't have time to go over it, but it turns out, and this is beautiful work done by a student jointly with Colleen Grill-Spector, that's Don Finzi, that the self-supervised TDAN model is a much better descriptor of the multiple pathway system, right? Suggesting that actually distinct visual streams at a much higher level can just be captured simply by learning a general visual representation in the context of a spatial constraint. So it's basically saying our argument is functional performance and spatial smoothness explains lots of data, like perhaps most of what's known about the visual system. But interestingly, there's nothing specific visual about that, right? The contrastive loss is basically independent of any kind of specific visual context, right? It's just visual because the inputs are images, right? And the spatial loss is nothing specific visual about that. That's just a simple biophysical principle, right? So what this suggests is maybe there's a general theory of functional organization here behind just like, beyond just something about vision, right? So maybe there's just speculation. Functional organization in auditory cortex, which is present both in earlier auditory cortex where you have tonotopic stripes, and higher auditory cortex where you have these other areas. Geeks on auditory system will tell you about those. Maybe this idea would describe those. And maybe even a wilder speculation is that the same theory will explain structure outside of sensory processing. So in like navigation and memory areas such as medial entorhinal cortex, hippocampus, where there are spatial functional organizations, perhaps they arise for similar reasons, okay? I'll just close by an attempt at trying to take models like that and think about them as having consequences for some further down the stream health application. So what would you do with a visual with a model of this kind, right? Let's say you're a patient that doesn't have sight but you want to use a model and some form of device to induce a percept, say, of letters, okay? Imagine you take that image of those letters, pass it through a network, right? If you pass it through a network, you get some form of activation pattern that arises. Okay, I'm just showing you the activation patterns for that image, right? Of course, if you're gonna use, say, stimulation to try to get the unseen person to have the percept, you're gonna use a device, and that device will have some kind of limited spatial resolution. So you're not gonna be able to induce exactly the internal representation that the original stimulus might have done, but instead it'll be like a noisy or blurry version of that, limited by how spatially diffuse the ability of the device is, right? If you induce it with a certain amount of noise, it might induce an imperfect, if perhaps acceptable percept. So this is like basically the image that would have generated that noisy pattern, right? So you might think, okay, well, if the goal was to generate a clean thing, this is what the percept would be if you have a noisy thing, if you were to have the amount of noise shown here in your device, okay? But different amounts of noise and different abilities to localize your stimulation to different areas have different types of percepts. So for example, if you're only able to stimulate in the early layer, early visual cortex, as the amount of noise increases, you get much worse ability to make percepts that make sense. On the other hand, as you are able to stimulate in more different brain areas, what you can do is trade off noise for different, for stimulation area. And it turns out that there's like a perceptual acceptability frontier that allows you to say, even at, say, noise levels that would be terrible if you could just stimulate in early layers, it's okay if you have that noise level, but you get to stimulate in several different areas. Right? And the key point is that you can only generate predictions like this using a topographical model, because this is the model that allows you to actually say how the interaction between diffusion in of the injected signal and percept actually would be. Right? And that's where this plot comes from. This is basically like a proof of concept of using models that are topographical and also functional to do better prosthetics design. Okay? So I'll just stop by saying that the contrastive approaches have largely made up the supervision gap, make a lot more sense in the when spatial phenomenology is taken into account. And there is a kind of unified mode agnostic principle to explain lots of data. So this is where we are in our report card. I will not mention learning rules today. That needs a whole other talk and is still sort of on very shaky ground. But I would say that the sort of general project of trying to improve neural models using advanced things in AI has been quite powerful over the past few years. There's also the other direction of using cognitive science to help AI, which is, again, another talk entirely. But I'll just put that slide up for a second since it's our latest thinking on it. Anyway, this is what we do in our lab, these 3 different approaches. And thank you to all the sponsors. And of course, the people who actually did it are the ones who should get the credit. So thank you.
So, okay, yeah, thanks, Dan. He squeezed quite a lot of information in this 45 minutes. But there must be questions, things that you probably didn't fully understand, or where you would like to know a little more about the background. So please, the audience in the room first. Yeah.
Hello. My question is, well, 2 questions. What was the total, would you say, aggregate of your research as far as years? How many years did you put into this so far? And then what would you say is the public's overall perception towards this field of work, and what are some common misconceptions that you could talk about? Thank you.
Oh, those are interesting questions. Not exactly science questions, but well, I don't know. We've been working on that for I tried to give you a history going back 10 years and describe how the lines of research have worked out over that period. So I've been at Stanford for 7 years, so that's roughly things we've done on the sort of computational neuroscience side over that period. I hope it was worth the time. I don't know. In terms of the public's perception, I don't really know. I often find myself surprised about what people say they think. So I don't really know what people think. I think most people don't think about that at all, probably. And then I know there's a lot of concern about AI generally. I'm not sure I'm addressing the question. I'm not sure I have a lot to say about it, but
No, this is great.
yeah.
I appreciate
Yeah.
whatever you share.
Yeah. Thanks.
Dan, hi, good to see you again. Couple of related questions. Have you done any TDAN type work with transformers? That's obviously not relevant or not obviously relevant in the same ways for making predictions about brains, but seems like it could be relevant for increasing efficiency of hardware implementations of transformers, and this is a place where maybe neuroscience could come back and have practical implications once again for AI.
Yeah, no, fair enough. I think it's a great question. Actually, we have. So what is the big, so people know about transformers, but transformers are not one thing. So transformers are different depending really on what the tokenization of the data is. And so in the case of vision, the natural thing is so-called vision transformers are to tokenize the data by making little patches. Okay, that's similar in the auditory data domain. Okay, so actually what happens is that you sort of have typically a positional encoding of those tokens, and it's done at either one or several layers along the pathway. And then there's a kind of one-to-one correspondence of the tokens at each of the layers of the deep transformer. So actually what you end up with is something that could look fairly convolutional depending on what's learned. And the big difference between a vision transformer and a convolutional network is that there's not weight tying in the same way in the vision transformer, and also there's instead of just things being local, there's the potential for after the first layer, long range attention to apply. Okay, it has been shown, not by us but by others, and we confirmed this, that very well trained transformers actually have a fairly local structure. Well, the well-trained vision transformers actually have a fairly local structure, meaning that if you look at the prevalence of the long-range attention throughout the layers, it starts out fairly local actually at the big early layers, and then you get more and more not long-range structure as you go by, as you go up the layers. But it's not entirely local the way it would be in a convolution network. But it's not entirely local in the real brain either. Okay, there are long-range connections even in early visual cortex. And so it's quite possible that the vision transformer architecture is kind of a generalization in some sense of it's not precisely, but something like, when trained, a generalization of the convolutional structure to have mostly local connections, but then some long-range connections as well. So that's a long preamble to the idea that yes, when you apply that same context in the context of the spatial constraints as well, which I didn't show, but that is work we've been working on. Indeed, you get even better models of topography, and that suggests that what ends up happening is that the spurious long-range connections that exist to some extent in non-topographical transformers can actually be suppressed out without any harm for performance, which is exactly the notion you were saying. So. Yeah, I think that's an idea that we think is quite promising, and hopefully we can actually explain what are called sort of non-classical receptive field effects by those somewhat sparse but meaningful long-range connections. Sorry for the long answer, but
yeah, that makes sense. Yeah. So more questions in the room here. Jan, do you have already something from our online audience?
A lot of people applaud online, but I don't really have much questions from online so far. So maybe I take this opportunity to ask a question myself. So then you're talking about unsupervised method, and also you analyze video recordings and from those cases, and how you put how do you kind of consider it the multimodality and in one model and have visual and the language and the environmental information analysis in one model. Yeah.
Well, so I mean, obviously the models can be multi-sensory, right? You can have audio-visual as well as audio. But are you asking about how Maybe
how
let
you
me,
integrate and all those modalities, and so yeah, came up to the outcomes.
I mean, I think that that I don't have to be creative to answer that question, since the field has been so generative at, no pun intended, at using multiple data. So, I don't think that there's necessarily any special sauce for the integration that's any different from the way multimodal models are built for the general AI case. Now, whether those describe multimodal integration in the real brain, I don't know, but it's an interesting question. And I think I have seen some works that purport to evaluate that question, although I don't know what the, and the results seemed reasonable, but I think I don't know that, it's not like off the top of my head I feel like there's any particular result there that's striking. It's not like there's some striking difference or a puzzle about multimodal integration that was like surprisingly explained or surprisingly failed or anything like that. Yeah, I don't have that. But again, that could be due to my lack of knowing what the situation is.
Thank you.
Yeah.
At the moment, you are using data that are being generated by neuroscientists, and the data they produce are usually because they have something completely different in mind. So they were solving all certain visual cortical problems, and they were using certain arrangements. So imagine you were in the driving seat. What kind of experiments would you suggest neuroscience should do to push it forward?
Well, actually, much of the data that I was described was developed in part by groups like Jim DeCarlo's, where he was motivated to try to produce data sets that were good for separating models. But and then some of the MRI data came from Kendrick Kay, who's built this natural scenes database, which is basically human recordings on MS COCO images. And those were well designed. Again, they were not, so I would say that like the main thing is that the main principle of design of the data should be separating models, like rejecting more models, having power to distinguish correctness of models, or make it clear where their errors are. So what that usually boils down to for me is very unclever experimental designs. So that's to say, just rectangular data sets that have like lots of stimuli and lots of neurons, or lots of voxels or whatever, and cover a wide range of stimulus types, whether they are kind of artificial ones like stimulus grading, or drifting gratings, or like natural scenes or whatever. Actually, it's good to have lots of natural scenes. I think that probably opens things up a lot. So basically, the idea is like record from real experience with as little artifice in the experimental design as possible, and that's actually the best case scenario, right? So,
yeah. So, if there are no more questions, I think we should thank Dan again, and then, yeah.